This unit was about DNA. DNA stands for deoxyribonucleic acid, and in which case it's going to be in a double helix twisted ladder-like structure. You'll find DNA in the nucleus of every single one of your cells, and it stores your genetic information. The smallest part of DNA is a nucleotide. There's millions of nucleotides in your DNA. They are comprised of a phosphate group and a deoxyribose sugar that makes up the backbone of your DNA. These will be held together by phosphodiester bonds. And then on the inside, or the ladders of the DNA, you'll have one of 4 different nitrogenous bases that will be held together to each other with hydrogen bonds. The 4 different bases will follow base pairing rules based off of Chargaff's rules. Adenine will always bond to thymine, and cytosine will bond to guanine. It's because of this that their percentages will always be the same. If there's 20% adenine, then there will be 20% thymine, and that remaining 60% will be split evenly, 30% for cytosine and guanine. DNA needs to be in every cell. So in the S phase of the cell cycle, we go through DNA replication, in which case we are going to have topoisomerase. An enzyme is going to relieve the supercoiling of DNA wrapped around histones. Then helicase will break apart the hydrogen bonds in between those nitrogenous bases. As we continue to split this, single-stranded binding proteins will attach to both sides of the DNA's replication fork to prevent it from reannealing or coming together. On the leading strand, or the original 3' end, we will copy continuously, starting with a RNA primer made by RNA primase. Then DNA polymerase will make DNA from the 5' to 3' direction continuously, following helicase. The lagging strand is a little bit more complicated. Because of the fact that we can go 5 to 3, we're actually going to be going away from the mouth of the DNA replication. So what will happen is we will add a primer, add DNA, add a primer, add DNA. And because they are not connected, these will form Okazaki fragments, or like unconnected pieces of DNA. Ligase will follow that newly synthesized Okazaki fragments on the lagging strand and fuse them together, fixing those phosphodiester bonds and making our DNA whole. This process is almost perfect. It's really, really good. But when it is not perfect, we have a mutation, which is a change in the genetic information. It's when DNA polymerase is copying, and instead of going C to G, it makes a mistake in one way or the other. One of those mistakes is called a substitution mutation. When your original DNA is CTT, CTC, and the new DNA is TTT, CTC, that is a point mutation, that is a substitution. This will change the protein in essence. A insertion mutation is when you add a nucleotide, thus shifting the rest of them over, and a deletion is when you remove one, thus shifting them the opposite way. Both of these are considered frame shift mutations because they're not only going to change the amino acid at the point that they happen, they will change the amino acid for every one after that. A special type of mutation called a silent mutation is a substitution point mutation where the actual change in the DNA doesn't affect the amino acid or the protein. All of this is just to get DNA in every cell. The reason we have DNA in every cell is explained by the central dogma of biology, which in the simplest form, the central dogma of biology is DNA transcribes to RNA, which translates to proteins. The way that this happens is that in your nucleus, there are going to be genes or sections of DNA that will code for proteins. You will copy that gene with RNA polymerase. That's going to open up, bind to, and make RNA, specifically mRNA. Once you have that mRNA, you will use SNRPs, or small nuclear ribonucleoproteins, to cut out introns, which are non-coding parts of mRNA, and you will leave exons, or coding parts of mRNA in. You'll fuse these together with the same enzyme from here, ligase. After you do that, you will add a poly-A tail, a bunch of adenine on the 3' end, and then a guanine triphosphate cap on the 5' end to protect it. Then you're able to leave the nucleus, feed through the ribosome, read the codon on the mRNA, which is a 3-letter sequence that will make proteins, attach the anticodon to it, and begin chaining together amino acids with the help of tRNA. That chaining together of amino acids is how we make proteins. We use all 3 different types of RNA, in which case they're all single-stranded, have ribose and uracil instead, and those are the main differences. Go get